Hey folks, Doug Blake with Body Design University. In this video, I want to pull you into chapter 5 because I do get a fair number of questions asking to simplify or explain in a more simplified way the sliding filament theory. It's kind of the go-to way of understanding how muscles function, work, develop tension, power, strength. And so in chapter 5, and I'm on page 145, it's a lot of information. So just a couple of things before I get started on here. It's kind of ironic, but one of the most important components of muscle physiology from a training perspective, i.e., exercise science, is to understand the concept of the sliding filaments, the actin and myosin, and how they slide across each other within a myofibril, within a muscle cell to generate tension. And that's the whole point of understanding the sliding filament theory. The other thing to kind of keep in mind, and that's why it's kind of ironic, is NASM is probably not going to ask you a ton of questions about this particular concept in specificity. It might ask you questions related to it, but at the end of the day, you're probably not going to get a whole lot of questions on the exam related to very specific components and elements. Now, you do need to know, for instance, that a muscle belly is comprised of fascicles. Fascicles are comprised of muscle cells or muscle fibers. Muscle fibers are essentially composites of what are known as myofibrils. Myofibrils themselves are where we see all the sarcomeres, and the sarcomeres, then if you broke them down and looked deep inside, you would see the myofilaments, right? The actin and myosin. And just again, keep in mind that from a purely testing perspective, certain things you do need to know, obviously, and I always recommend that you memorize the terms on the side panels. Excuse me, of course. Anytime you see numbering and any type of listing of information, you definitely want to go in and kind of look to memorize that information. Of course, if you see any tables, any charts, as I'm going through this, I'll notice table 5.3 and all these other tables. Those are generally where you want to put the majority of your attention. But again, in chapter 5, because there's a lot of information in here, you got to be careful that you don't spend too much time, because again, it's not like you're going to get 10 questions on the sliding filament theory. Not that I've ever seen or heard students remark about that. On the other hand, know it so that you get an appreciation for the way muscles work to develop tension. And that's really what NASM is trying to do. And I think they did a great job in kind of paring it down to the level that a personal trainer needs to know this information. Of course, if you want to delve deeper into it, because it's a fascinating field of physiology when you go deep into the muscle and the sarcomere and down at that level where you see all the proteins that are involved in holding sarcomeres together. Very cool. But again, in this video, just want to kind of give an explanation of the sliding filament theory, in particular the way NASM explains it to you. A muscle contraction, shortening of a sarcomere. What is a sarcomere? The general definition of a sarcomere is that it is the fundamental contractile unit of a muscle cell. Okay, so we always think about muscle cells, these very elongated cells in the body that are connected on either side by tendons, in general by tendons, which attach to bones. But we seldom dive deep into exactly what's inside a muscle cell. These are incredible and incredibly complex machines. Okay, so you've got to scratch below the surface, and when you look inside a muscle cell, you see a bunch of different things, lots of nuclei, right? Because that's where the DNA is that's going to code to create the proteins necessary to build, grow, maintain the muscle fiber itself. But if you scratch deep down enough, you're going to see these sort of banded fibrous components. And if we look down at it, we can kind of isolate the larger components, the myofibril. But when you go down deep into it, you can see these things, and we call them sarcomeres, right? So you have these individual units. And of course, there's thousands, tens of thousands along a single strand of muscle fiber. And those are called sarcomeres. So it's the sarcomeres that contain the actin and the myosin. Those are the basic contractile myofilaments. We call them contractile. Technically, we would just call them myofilaments that act as tension development devices. So sarcomeres are in series from one end of the muscle fiber, depending on the type of muscle it is, to the other side. Tendons hold everything to the bones, and so when you think of it from that perspective, you can understand it's pretty complicated. We got to scratch way below the muscle, down below the muscle cell itself, and look deep into it, and you're going to see what we have here on page 145, and that's figure 542, sliding filament theory. It's not a bad way of understanding it. Of course, there's significantly more complicated processes, but the most fundamental process is is how the actin filaments on either side of a single banded, it's actually a bilateral band of myosin filaments that have what are known as heads on either side. So an actual myosin protein, pretty big protein, but it essentially has it's a unilateral type of protein. So it has a tail end and a head. So if I put one with the tail end here and the head on one side, I can now mirror that with another one with a tail here and a head here. And now you have what we call a myosin myofilament. So let me say that again. The actual myosin protein, it's a big protein, by the way, large, lots of amino acids that make up a myosin protein. So those proteins have a unilateral dimensionality to them, meaning they have a tail, whoosh, and a head. And so if I grab a bunch of those, like think about grabbing like a bundle of I don't know, name your favorite herb. I'll use parsley if you ever seen parsley. You can grab a bunch of parsley. So you got the stalks and all the bushy heads on it. So if you take that to one side, that's kind of like what you would see a myosin filament. All of these myosin proteins that are unilaterally um, dimensional. So one side has one component and the other side has the other. It's the easiest way to think about it. Now, so you got that one bunch of celery, stalk and the heads, right? And those heads you can kind of think of as the myosin heads. Take another stalk. So you got these two identical stalks, right? Stalks down at the bottom, stalk down at the bottom, heads at the top, heads at the top. And now go like this, like putting like this together. And now you've got these bushy heads of, I'm saying parsley, on either side, and then the stalks go in the middle. That's one of the easiest ways to kind of visualize what we're talking about here with myosin molecules. And that don't take it any further than that. It's just that these are the proteins. They have a central location that are held together by other proteins, and they're able to pull and grab onto and pull the actin filaments together. So with that said, that then becomes sort of the basis for what is known as the sliding filament theory to produce tension. Now, remember, that's a single sarcomere, but there's multiple sarcomeres within each myofibril because essentially, without getting too deep into it, a myosin actin coupling scenario is a single myosin with 6 actin proteins or actin filaments surrounding that single myosin myofilament on either side. So again, we're looking at things in 2D, but really in 3 dimension. You can Google this, by the way. Just Google 3D sliding filament theory. Got some great, I mean, there's a ton of great material for visual effects. But that's only if you want to scratch way below the surface. It's not necessary from a testing perspective. But if you do, it's really cool to see it's a hexagonal structure with a single myosin filament and 6 6 around it. And so 6 actins that actually get connected to by the myosin, and the myosin heads pull, flip, do these other things. And that's what you can read here on the side panel. So for the most part, you're getting all you need to know about the sliding filament theory on the page 145, the side panel. Sliding filament theory, what is it? Produces a muscle contraction, shortening the entire length of the sarcomere, all the sarcomeres shortening at the same time, the entire muscle fiber within, or the myofibrils within the muscle fiber, They all shorten at the same time. Get that over thousands and tens of thousands, maybe even 100,000 plus muscle fibers, and the entire muscle belly can shorten. Really pretty cool. The next one here, and I'm just going by the way I would want you to study this. Excitation, contraction, coupling. Again, don't let the terms throw you off. It's just a process where the electrical stimulus from the nerve cell, right, from that efferent nerve, right, sends an impulse from the axon. The axon, remember, in skeletal muscles, the nerve body of that nerve cell with its exceptionally long axon, the nerve body resides in the spinal column. Okay, so the initial impulse, that initial action potential that's generated at the nerve cell body sends the impulse down the axon of the cell, which goes all the way out to its target effector, and in this case, it's a muscle. So an effector can be any organ or that a efferent nerve goes to stimulate. In this case, it's a muscle, and so the muscle fibers receive this electrical stimulus. That's what's called the excitation. Ooh, exciting, electrical. Think about electrical excitation, contraction, coupling. This occurs, boom, and this occurs. And that's what it's saying. Physiological process of converting an electrical stimulus to a muscle contraction. That occurs really sim. By the way, they give you a picture, by the way, figure 541, because this conversion of electrical stimulus to a muscle contraction, that intermediary conversion process is an electrochemical. So you got an electrical stimulus, which then causes chemicals or neurotransmitters to be released at the what we call the synaptic cleft, right? And so that allows for the movement of neurotransmitters onto the muscle cell. If the excitation or the electrical activity is sufficient, okay, it will cause calcium ions, which are located within the endoplasmic reticulum, to be released. Calcium floods the muscle cell. It's calcium is the key. I'm not going to go into it any deeper than that, but you can look at it, you can read it. But calcium is the key, because without it, you can't have the displacement of the um, component of the actin filament to move away, allow for the myosin head to attach to it, yada, yada, power stroke. And that's what you're seeing here. Excitation, contraction, power stroke is the myosin heads bind. And this is where, again, like I'm saying, it's not a bad idea. If you want, you can look up any of these 3D visuals, and there's some really cool visuals out there that you can look up video wise that will give you a better appreciation for it. Myosin heads bind to actin, pull them towards the sarcomere center, which slides the filaments past each other, shortening the muscle. Before we go on, if you find this somewhat boring, I get it. Let me get you a little bit more interested in this. All of this occurs in literally hundredths of a second. This is one of the things that I've always found incredibly interesting and made me even more interested in trying to understand this. Because once I understood the time frames that we're talking about like that, faster than that. Okay. You know how when you're watching like the Olympics and you see runners and some guy wins, on the 100-meter, some guy beats another guy. First and second place can often be the difference between 1/10 of a second or 5/100ths of a second, a half of a tenth. I mean, it's bizarre. You look at them and it's those kind of time frames that we're dealing with. It's actually faster than that. Because if you remember the speed of the action potential down the axon of a nerve cell is a whole lot faster than anything you can even understand or imagine. The things that are occurring at the level of the actin and myosin heads, the myosin head and the actin filament, these are occurring in hundredths of a second. That's how fast this stuff is happening. So in order for you to literally contract your bicep, there's literally millions of engagement actions occurring like that to make that happen. It's mind-blowing when you think about the complexity of just a human muscle cell and the sarcomeres. But I think that's important to kind of keep that in mind, because I kind of you go, what? 10th of a second, hundredth of a second. By the way, you don't know what a hundredth of a second is. It's not anything that is real to us. It's more academic than anything else. It's real in that it happens, but our ability to comprehend that, it ain't happening. So you just got to know it. So the power stroke is the myosin heads bind to axon, pull them towards the sarcomere center, which slides the filaments past each other, shortening the muscle. Remember, that's in every sarcomere, in every muscle cell that's actually providing the shortening. You seldom, if ever, get 100% of all of your muscle fibers working at one time to produce movement in a particular muscle over a particular joint. It just doesn't make sense. You don't need all that power from most 99.9999999% of the time. And by the way, if the average person was able to excite all the muscle fibers and get all the muscle fibers at one time to contract against the load, you'd probably injure, rupture the tendon or tendons, or even rupture some of the proteinaceous components of the muscle fibers themselves. You'd hurt yourself. That's what it comes down to. Adenosine triphosphate, of course, is ATP. That is the molecule of life, obviously energy molecule. All life on Earth uses it. And that serves as the main form of energy in the human body, known as the, okay, they're just giving you that as a, reiteration of what we already know. ATP is what allows for the sort of physical manipulation of the actin myofilament, because there's multiple components, proteins in there that are actually able to twist, move away, allow for the myosin. ATP is also responsible for getting the myosin head back to its normal position. Not really that necessary to note, but it is important to understand, obviously, ATP, because they will ask you some questions on that. Resting length of a muscle when it's not actively contracting or being stretched. So there is the basic outline of the sliding filament theory. And now and at the bottom, they give you basically a 5-step process, which is not a bad idea to at least read it and understand it. The steps in the muscle contraction, called excitation, contraction, nerve impulse begins, central nervous system, right? That's occurring in the spinal column. Now, this is generally understood, right, to be sort of the main component, the central nervous system. So central nervous system, cerebral cortex. Whoop, I want to contract my bicep is what I'm thinking. That nerve stimulus goes down into the spinal column. There's a couple of intermediate steps, and then the axon that connects all the way to the muscle is going to provide the action potential. Acetylcholine, so the action potential, bam, that electrical activity right at the synaptic cleft, released. So acetylcholine is released into the neuromuscular junction. You got that right here on page 144, which then helps the nerve impulse cross the synapse into the muscle. That's what that's known as, the synapse. Nerve impulse travels into the muscle infrastructure, stimulating a small organ called the sarcoplasmic reticulum to release the electrolyte calcium. I already explained that to you. Calcium is then released into the muscle, floods the entire muscle, stimulating a chain of events that results in the myosin heads binding to actin. So literally, number 4 just essentially told you that there's about 20 or plus reactions that are occurring like that, just to get the calcium to do what it does, stimulate a chain of events. By the way, that's why I'm saying it doesn't make it's not important that you know any of those events, to be honest with you. Even this is more than you need to know, but you do need to understand just the sliding filament theory. This is about as simple, as basic as it gets. Myosin heads then pull the actin towards the sarcomere center, slice of overlapping filaments past each other. So this is your basic outline of the sliding filament theory. They do give some good pictures. And by the way, 2D photos, that's fine. But like I say, it's 2024 when I'm taping this. You have access to incredible 3D stuff you could watch. Again, I only recommend that if you're interested in more sort of in-depth information or in-depth knowledge, visuals that can help that are not going to help you on the test because they're just not going to ask you for the most part. And if they do, it's a question or two. I don't even think um, in the most recent exams that they even have stuff like this on there. So chapter 5 becomes one of those chapters where it's just my opinion. I think it's important that any trainer should know some of the, some of the more foundational elements of muscle physiology for sure. But other than that, you're now going to move on to the motor units, all or nothing principle, and then ultimately into fiber type. So at the end of the day, learn these components of it, meaning more superficial elements, and these terms on the right side of page 145. But again, from a testing perspective, not a whole lot more than that. But if you really want to know it, which I would say is kind of cool, and if you're a trainer, you're probably really into sort of the exercise sciencey, academic-y stuff. At least I would hope you would have some interest in that. Then go dive a little bit deeper into it. But I'm hoping that this video was helpful in kind of giving you a little bit more flavor of the sliding filament theory. At the end of the day, neurological stimulus creates a chemical cascade of neurotransmitters, release of calcium floods through the sarcoplasmic reticulum. Those calcium ions are the key to the structural changing shape of the actin. Now the myosin head can attach to it. You have ATP in this process to ensure all of that happens, plus the myosin heads backing down, going back to their normal resting length. It's that attachment of the myosin head and the power stroke of the myosin head on either side of the myosin filaments. Boom, those heads grab onto the actin, pull them forward, and that's how muscles generate tension and then ultimately shorten. And there's your sliding filament theory. So if this was helpful, please write the word understand in the comment boxes. Remember to give us a thumbs up. Share it if you want to as well, and make sure that you subscribe to the channel. If you got any questions, please leave them in the comment box so we can reach back out to you. Have a great week. We'll see you next week.